Okay. Hi guys. My name's Astrid. So I decided to write about prompt number 4, which is about lipid digestion and absorption. So consuming a fatty acid like an avocado or MCT oil, the body processes the long fatty chain acids and medium chain triglycerides in different ways due to their chemical properties. Um, I'm going to be talking about the hydrophobic and hydrophilic natures of the lipids, the enzymes, and the hormones that aid into the digestion of it, um, which is here. All right, so properties of lipids. As shown in the picture, you can see the hydrophilic head, which is on the outside, and then the hydrophobic tail is on the inside of the micelle. So lipids are largely hydrophobic, meaning that they do not dissolve in water. This property leads them to form large globules in the watery digestive tract. However, some lipid molecules contain polar head groups that can interact with water, giving them limited hydrophilic properties. This dual nature requires biosalts and carrier proteins to assist in the digestion and absorption of these things. So again, hydrophilic, water attracting, hydrophobic, water repelling. Um, Emulsification by bile is required for efficient digestion, and amphipathic nature explains why lipids require a special transport. So enzymes in lipid digestions. We have lingual lipase, gastric, pancreatic, and cholipase. Um, I'm sorry if I mispronounce words, but so lingual lipase is secreted by the mouth, minor digestion of triglycerides. Gastric is secreted in the stomach and works in acidic conditions. Pancreatic lipase secreted in the small intestine, which is the major enzyme. And then cholipase, it stabilizes pancreatic lipase on fat droplets. So lipid digestion begins with lingual lipase from the tongue and the gastric lipase from the stomach. However, the majority of lipid breakdown occurs in the small intestine by the pancreatic lipase secreted by the pancreas. This enzyme hydrolyzes triglycerides into monoglycerides and free fatty acids. Cholipase, also secreted by the pancreas, stabilizes pancreatic lipase, ensuring effective fat digestion, which is all shown in the picture here to the left. Hormones in digestion. So we have CCK, which stimulates bile release and pancreatic enzymes. We have secretin, which stimulates bicarbonate to neutralize acidic chyme. We have GIP, which is gastric inhibitory peptide, which slows gastric emptying for better absorption. Several hormones coordinate lipid digestion. CCK is released by the small intestine, stimulates gallbladder contraction and pancreatic enzyme secretion. Secretin promotes bicarbonate release to neutralize acidic chyme, creating a suitable environment for enzymes. Additionally, hormones such as GIP and GLP-1 slow gastric emptying, giving the intestine more time to process dietary fats. Right here for bioproduction, oops, right here. Okay. So bile is produced in the liver and stored in the gallbladder. It is released in the duodenum upon fat ingestion. Bile salts are amphipathic, which means they emulsify fats into smaller water droplets. And then emulsification increases surface areas for enzyme activity. Here's a pretty picture that I found. I have my sources at the end. Just showing the whole traction of bile, where it's made in the liver, it's stored in the gallbladder, what it helps with, and then what happens uh, when it starts to come out, if that makes sense. So again, bile is produced in the liver from cholesterol and stored in the gallbladder during digestion. CCK signals the gallbladder to release the bile into the small intestine. Bile salts emulsify fats into smaller droplets, increasing the surface area for pancreatic lipase to act. Most bile salts are reabsorbed in the ileum and recycled through the enterohepatic circulation, while some are excreted in the feces, helping eliminate cholesterol. Function emulsifies fats. Recycling, most bile salts are reabsorbed in the ileum, liver. Excretion, some bile salts leave via feces, helps remove cholesterol, and bile salts are critical for both digestion and maintaining cholesterol balance. Okay, lipid absorption. Once fats are emulsified and broken down, micelles transport fatty acids and monoglycerides to the enterocyte brush border. Inside the cells, long-chain fatty acids are re-esterified into triglycerides and assembled into chylomicrons. These particles enter the lymphatic system through lacteals before eventually reaching the bloodstream. Medium-chain fatty acids, by contrast, bypass this process and enter directly into the portal vein bound to albumin, traveling quickly to the liver for oxidation. So micelles carry fatty acids and monoglycerides to enterocytes. Inside those enterocytes, fatty acids and monoglycerides, which are re-esterified into triglycerides. Triglycerides are packaged into chylomicrons or transported directly depending on the chain length. And then micelle-mediated absorption is essential for hydrophobic molecules. For the long chain fatty acids, they're packaged into chylomicrons. They enter the lacteals, like I just said, then to the lymph, then to the bloodstream. There's slower energy release compared to the MCFAs, and then the major pathway for dietary fats and avocado and most natural foods. And then the medium chain fatty acid, it's absorbed directly through the portal vein. Is transported quickly into the liver and then rapidly oxidized for energy. Doesn't require chylomicron formation or lymph transport, and it's ideal for ketogenic diets or quick energy needs. These are my sources. Thank you very much.